I'm Jenna Mattice. I am a PhD student in chemistry and biochemistry, and today I'm going to tell you about carbon dioxide and enzymes. A love story. So, carbon dioxide is at historic highs. Uh, Even though the Earth normally goes through cycles, since 1950, it's really just a ridiculous amount of carbon dioxide in the atmosphere. And as we think about, what that means for us, what that means for our life on Earth, I want to tell you a little bit about the natural processes that capture and fix carbon dioxide. So the big ones are in plants, right? That's photosynthesis. So just like this field of poppies, they're breathing in carbon dioxide and they're making it into little plant parts or little poppy parts, right? Uh, but besides plants, other organisms do this too. And another organism that does, or another set of organisms that do this are microbes. And so microbes also breathe in carbon dioxide, and make it into little microbe parts. And plants and microbes, they do this for very different processes, but they all utilize enzymes, sometimes called proteins, but they're biological molecules that really help chemical reactions happen. And so I kind of said this already, but they're all doing this through different enzymes and different processes, but they all have to kind of overcome the same challenges in order to bind carbon dioxide and make it into biomass. And these three big challenges I think you'll find are the same as if you choose to online date. So if you're online dating, your first challenge is that you have to capture that person's attention, right? And that can be kind of hard. I mean, there's a lot of people out there. And the second challenge is after you capture someone's attention, how do you discriminate between whether or not someone is cool or someone is a jerk? So discrimination is a big part of this. And then number three is you have to bond or you have to connect with them. So you want to I'm assuming meet up for a date, or you want to have some good conversation at least. These are really important. And it's the same thing for the enzyme. So the enzyme, number one, when he's looking for carbon dioxide, actually has to capture it. And that can be really hard because there's a lot of other gases out there. And carbon dioxide only makes up about 0.03% of the atmosphere. So just not that much. He really has to discriminate, right, between carbon dioxide and more abundant and reactive gases. And in the enzyme's case, if it doesn't discriminate between carbon dioxide and, let's say, oxygen, it's going to end up in not being good for the organism. So here I'm showing carbon dioxide. It's binding and combining to this substrate, and then it's going to be made into these products, which will end up making little poppy parts. But let's say that this enzyme instead reacts with oxygen. It will end up actually in the wrong products, and you're not going to get your biomass. You're not going to get your nice field of poppies. So discrimination is really important. And number three, the enzyme has to bond, actually make a bond with carbon dioxide. And so how it does this is by generating a nucleophilic carbon near the carbon dioxide, and then it will form this CC bond, right? So the three enzyme dating challenges are really the same as humans. So it's going to be capture, discriminate, and bond. And me as enzyme matchmaker, I really focus on discrimination. So how does that enzyme choose between carbon dioxide and something else that's more reactive and more abundant? So I want you to imagine for me, oh wait, I messed this up. So enzymes have a strategy where they will bind something else and they will change their structure. And through that, That's how they will bind the correct substrate and exclude something else. So in this case, we're binding carbon dioxide and we're excluding other gases. So now imagine for me that I have a bin and it has a bunch of different balls in it. So it could be tennis balls, it could be baseballs, it could be basketballs. And I said to you, like, hey, catch this ball. You're not going to really know what I'm throwing, right? You could potentially catch any of them, but you're not quite sure which one I would throw. If at first I handed you a baseball mitt, you would then naturally gear up for a baseball, though, right? You would ready yourself and you'd be like, okay, I'm going to catch this baseball. So if I handed you a mitt and I threw you a basketball, you'd be like, no, no, thank you, I don't want that. Why did you hand me this mitt? It would be very confusing. And so this is how the enzymes actually ready themselves as well. And, like all over the place. And so the big way that I study this is actually using mass spectrometry. So mass spectrometry is the mass to charge ratio of a molecule, and that will actually be the fingerprint of a molecule, is sometimes what it's known as. And you guys have actually all probably seen a mass spectrometry spectrum, because they're really common in like crime shows. And in crime shows, it's a little bit different, right? They like dim the lights, there's like suspenseful music, people are like squinting really hard at a computer. And for me, it's a little bit different. Whenever the lights are on, I'm like, I'm like, you know, maybe playing some music, I'm looking at protein stuff. So, how does mass spectrometry really work? Mass spectrometry, first you have to charge that molecule, so it's actually going to be ionized. Then it will go through a mass analyzer, which will then differentiate between different masses and charges before it will eventually hit a detector and produce this kind of spectra. So all of these different peaks in this mass spectrum are representative of different mass to charge ratios, meaning this charge and this partial part of this structure is one line. And from all of these peaks, we can come up with the total structure of this molecule, which is acetone. So there's nothing to it. For proteins, though, it can be a little bit more challenging. And that's because proteins have really complex structures. So all of these peaks are also representative of different mass to charge ratios for this one beautiful structure. And so what I do is I will poise that enzyme with different mitts or different chemical conditions, you know, in the presence of CO2, and I will see how this spectra changes. And from that, I can make real determinations about the protein structure and its movements. So, why is this important? This is so important because even though, you know, carbon dioxide has been really heavily studied, we don't know everything about it. And we don't know all of the chemistry and biochemistry that happens in the natural world in terms of this field of poppies, or you can think about crops. And then, of course, it has other applications when we're thinking about, you know, what kind of products can we make? What kind of mitigation strategies can we do as we move forward? So it's really exciting work. I want to say thank you. I want to say thank you to my lab and my funding, and I want to say thank you to STEM Storytellers. It's been a really, really great experience, and you guys are all amazing people. Thank you.